Thank you for joining us. All lines are currently muted, and we will be getting started shortly. Hello, everyone, and welcome to the Foundation Fighting Blindness National Chapter Vision webinar. My name is Amanda Bement, and we are so excited to be with all of you today, and thank you for sharing your Saturday with us. I would like to briefly review some logistic details for this call. Currently, all participant lines are muted. Today's call is being recorded and will be shared with all registrants after the meeting concludes. And you will also be able to find the recording on the Foundation Fighting Blindness website within the coming days. If you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. This control bar may collapse when not in use, so make sure to jiggle your mouse before clicking anything. If you prefer to prevent the controls from auto-hiding, you go to your settings within the Zoom platform. You can select Accessibility, and then select Always Show Meeting Controls. If you have questions throughout the presentation, please send them using the Q&A function or send to chapters@fightingblindness.org. Next, I would like to welcome Dan Day. Dan is the president of the Foundation's Orlando chapter and will kick off this webinar with some exciting updates and ways to get involved with the Foundation. Dan?
Thank you, Amanda. The Orlando chapter of the Foundation is pleased and honored to host this edition of the National Chapter Vision webinar series. Again, I'm Dan Day, president of the Orlando chapter and a member of the Foundation's National Board of Trustees. I have autosomal dominant retinitis pigmentosa, as do roughly 10 members of my family, ranging in ages from toddlers all the way to my mom, who turns 100 next year. We call ourselves the Orlando chapter, but in fact, we span quite a wide geographic territory here in Central Florida, with members from Kissimmee and Leesburg and all the way over to the Space Coast. I've been involved with the Orlando chapter for over 30 years now, and it's been my privilege to be the chapter president now for over 20 years. I'm also chair of the Orlando Vision Walk. In May of 2006, the Foundation chose Orlando as a site for its very first Vision Walk. And since then, we've raised nearly $1.5 million to support the Foundation's work. We're currently preparing for our 17th annual Vision Walk, which will be next March. We are actually one of the oldest chapters in the Foundation, having been created in 1978. I'd like to give a special shout out here to members Bill and Janet Chatlos, the Chatlos family, and the Chatlos Foundation. Bill and Janet were co-creators of the chapter back in 1978. I think many of you know that the very first Foundation-funded laboratory was the Berman Gun Lab in Boston, and that was led at the time by Dr. Elliot Burson. Dr. Burson was the first recipient of the William F. Chatlos Fellowship for the Study of Retinal Disease at Harvard University. The National Chapter Vision webinar series is actually funded in part by a grant from the Chatlos Foundation. So we are very pleased and grateful to have Bill and Janet as core members of our success here at Orlando. We want to, as a chapter, we want to extend an invitation to all of you to come join us next June here in Orlando at Disney World at the Coronado Springs Resort Hotel for the Foundation's Visions Conference. This will be on June 17th and 18th, which is a Friday and Saturday. The conference will bring together people with retinal disease along with researchers and clinicians to discuss the latest scientific breakthroughs, to talk about all of the great scientific research that's going on, and to talk about the great progress we're making with our clinical trials. There'll be Q&A sessions with doctors that you can attend. There'll also be sessions for coping and sessions about technology, and a very large exhibit hall that will have lots of technology, assistive technology for the visually impaired. So I hope you'll consider taking some well-deserved vacation next June and coming down to Orlando, enjoying the magic of Disney World and catching up on all the latest science at the Visions Conference. And most importantly, having the opportunity to reconnect with your friends and to make new friends at the Foundation Fighting Blindness. Registration for the conference will open soon, but in the meantime, you can find information about the Visions Conference at fightingblindness.org/visions-2022. So I hope you'll come and join us next June for the Visions Conference. I want to briefly mention today, too, the Victory for Vision campaign. If you haven't heard of Victory for Vision, that's our current campaign to identify $50 million over the next 5 years to support the Foundation's critical mission. One part of this campaign is called Transformational or Major Gifts, and it has a goal of identifying $20 million. Another division, of which I am honored to be a co-chair, is called the Riensma Legacy Society Division, and it has a goal of identifying $20 million. I am very pleased to tell you today that the transformational part has already exceeded 75% of its goal, having identified over $15 million in pledges. And I'm equally excited to tell you that the Riensma Legacy Society portion has already exceeded 100% of its $20 million goal. So we're very excited to tell you that, and very excited that we are well ahead of schedule on this campaign. But make no mistake, we are not resting on our laurels here. We're going to move forward aggressively to turn this into not just a victory for vision, but a blowout. So I mentioned now $40 million of this $50 million Victory for Vision campaign. The other $10 million will come from sources such as new donors and new events, such as Hope from Home and Music to Our Eyes, as well as a renewed focus on fundraising in our local chapter network. So I know that many people on this call right now listening in have already pledged to be part of Victory for Vision, and we can't thank you enough for your support. If you're wondering how you can be part of the victory, you can support these events that I mentioned, like Hope from Home and Music to Our Eyes, that you'll hear about in the coming months. And if you're not part of a local chapter today, I hope you will give serious consideration to joining a chapter. It's easy, doesn't require any large time commitment. And it's a great way to stay connected with your friends at FFB and to keep abreast of all the great science going on. So if you'd like to find out more about joining a local chapter, just go to fightingblindness.org and click on the local chapters link, and you'll find all the information there on how to contact your local chapter. So today we're going to be hearing from some amazing researchers on progress in emerging therapies for retinal disease. Our researchers have a wide breadth of knowledge in this field, and the Foundation, though we do continue to fund research for therapies for specific gene mutations, we maintain a very strong focus on what we are referring to often as gene-agnostic or mutation-independent approaches that can apply across the broad spectrum of retinal disease. And these approaches hold out great hope for people of all ages and in all stages of vision loss. So the Foundation at the moment is funding more than 40 clinical trials. And in fact, I think we have are zeroing in pretty quickly on 45 clinical trials, with that number growing ever more quickly. So I hope that when you've heard from our researchers today that you'll agree with me that though we have been called the Foundation Fighting Blindness for a long time now, today we aren't just fighting blindness, but together we're winning. So let's get on with the science. To kick it off today, I'm pleased to introduce Ben Shaberman, our Senior Director of Scientific Outreach and Community Engagement. For over 17 years, Ben has been reporting on the research advances in the Foundation Fighting Blindness through both print and electronic publications. He gives presentations on science and research to the local chapters of the Foundation, as well as external organizations that are involved in supporting the visually impaired community. He provides training sessions to the foundation staff and to volunteers so that we can help spread the word in our local communities about the great research the foundation is doing. He also works with constituents to help them find the areas of most promising research for their specific retinal diseases. And in addition, he is the host of the informative Eye on the Cure podcast. And if all of that isn't enough, Ben is an accomplished writer, having published the acclaimed science fiction novel Retina Boy, available at Amazon in paperback and Kindle audible formats. That would be the perfect holiday gift. So Ben, I'll be looking for some kickbacks on those royalties there. But actually, we are very, very fortunate indeed to have Ben as part of our foundation family. So take it away, Ben.
Well, thank you, Dan, for kicking off the program and the introduction and, of course, the book plug. I wasn't expecting that, so thank you. And Dan, you really are a superstar when it comes to fighting blindness. I've known you since I've been with the foundation, and I was looking in our electronic records that really only go back about 30 or so years, and you were there at least when we began keeping electronic records for those 3 decades and maybe even longer. So thanks for all you do. You help us drive the mission in so many ways, including with my little professional outreach team, educating eye care professionals about the foundation. And I want to thank all who have joined us for this webinar on emerging therapies. It's really exciting to have a truly global audience. And we appreciate the many of you who support our mission in myriad ways as volunteers and donors, because at the end of the day, you drive the mission. And as most of you know, we're celebrating our 50th anniversary this year. And through your generosity, your dedication, your hard work, we've raised more than $850 million to support our mission to eradicate all the retinal degenerations. And I want to give a special shout out to those people in our chapter network, which is just growing by leaps and bounds. Our new chapter initiative is called Luli's Next Chapter, and it was named to honor the legacy of co-founder Luli Gund, who was so passionate about our chapters. And chapters give the foundation a really strong presence in more than 40 communities throughout the U.S., and it's often the first stop for people and families newly diagnosed with a retinal degenerative disease. And I also wanted to say a word about our website, something that's near and dear to my heart. If you like learning about the science and the research, and that's sort of a rhetorical comment given you are on this webinar, you really should check out fightingblindness.org for the latest in research, educational videos, information on genetic testing, and access to our Eye on the Cure podcast. And at the very top of the homepage, you can see this orange bar, and if you click on that, you can register for receiving updates through email on research and foundation news. So that and the website are great ways to stay connected to the research. And as Dan said, we have 4 presentations coming on a broad range of emerging therapies for retinal degenerations. And we will have time at the end for questions. You can enter your questions, as you heard, in the chat or the Q&A on the Zoom bar. But before we get going and I start introducing our speakers, I wanted to recognize our sponsors. And I want to bring up a slide here. And let me hold on one second. I want to get it in full screen mode here so you can see it well. I want to recognize our many partners who are so critical to making efforts like this chapter webinar possible. And they include Genentech, Janssen with Mira GTX, AGTC, Editas, 4D Molecular Therapeutics, Apellis, Astellas, Biogen, Spark Therapeutics, Boehringer Ingelheim, and Pixium Vision. And many of these partners also support other parts of our program. So thank you to them. Okay, so now it's my pleasure to introduce our first speaker, and that is Dr. Daniel Chung. Dr. Chung was appointed Chief Medical Officer of SparingVision in February 2021. In his role, he leads the clinical development and research into SPVN-06, and that's SparingVision's flagship development asset. And he's also responsible for the clinical development of current and future pipeline products. Dr. Chung also leads the education of the medical community and patient advocacy. And prior to joining SparingVision as their Chief Medical Officer, Dan led the global medical strategy for ophthalmology at Spark Therapeutics, a leading rare disease gene therapy company and a member of the Roche Group. And I might add, they are the developer of our first gene therapy, Luxturna. He played a pivotal role in the clinical development and approval of Luxturna. So Dan, the floor is all yours, and we look forward to hearing about SparingVision.
Well, thanks so much, Ben. It's really a pleasure to be with all of you today. And I congratulate Foundation Fighting Blindness for the 50 years of service they have done to the community. I know it's always been a pleasure to work with you, and that dates to the days that I was back at Spark and the University of Pennsylvania. So great to be with you today. So I just wanted to give you a little bit of idea of what SparingVision is. I know that this is a relatively new company and that some of you may not have heard of this, but we are what we call a genomics medicine company. So basically anything therapeutic-wise that we utilize the genome or the genes to look for a therapy for inherited retinal disease or other ocular diseases. And we are strictly in the ophthalmology space. We don't plan on going into other therapeutic areas, so which is quite fine with me because I'd be pretty much lost if we went to something other than ophthalmology. So with that, I wanted to mention that for our gene-agnostic part, because that is one of the things that we're really concentrating on, is that it really started with a long story. So the idea was that with our founders, Professor José Sahel and Thierry Livellard, who were at the Institut de la Vision in Paris, France, and Professor Sahel currently has moved on from there and is the department chair of ophthalmology at the University of Pittsburgh Medical Center. They asked this fundamental question, and that was concerning a very relatively common group of inherited retinal disease, and I know that it's still a rare group of rod cone dystrophies, one of those being retinitis pigmentosa. And if you remember, and I'm sure many of you know this, is that there are these cells in the back of the eye known as photoreceptors, which are these light-sensing cells. And there are two types. There's the rods that are in the periphery. They're responsible for peripheral vision and nighttime vision. And then there are these cones that are more in the central part of the retina, and that's where your daytime vision, color vision, and your fine acuity. Right now, a lot of the cones are really firing if you're able to see the screen. And so the idea was they asked this question that there are over 70 different genes that cause retinitis pigmentosa. And so we know why the rods die, because the genes actually affect the mechanisms in the rods. But the question was, why did they then go on and have this demise or this degeneration of the cones? Because there didn't seem to be a direct genetic link to that. So through their research, they found that there was a gene called NXNL1, or the nucleorhodopsin-like 1 gene. And in that, they identified this factor that they termed rod-derived cone viability factor. And just to keep this somewhat short, that factor they found was actually given off by these rod photoreceptors, so the photoreceptors that are in the periphery, and they actually had receptors on the cones. And that's how these cones really stayed viable and functional because they had this huge metabolic influence on the cones to help them survive and maintain their structure and things of that nature. And it was also a powerful antioxidant that also helped because the retina is so oxygen rich and that it uses a lot that you have to really take care of those what we call those reactive oxygen species that are detrimental to those cells. So the idea is with this metabolic effect and this antioxidant effect is how the rod cells support the cone cells. But in retinitis pigmentosa, all these genes cause the death and destruction of these rod photoreceptors. So the simple principle, and this is where the gene-agnostic part comes in, is that it doesn't really matter how or why those rods die. It's simply the fact that they're no longer there producing this RDCVF to keep the cones alive. So for us, we're trying to do gene therapy where we're actually replacing the RDCVF that is no longer being produced by those rod cells because they're not there anymore, and help support cone function. And so the idea is that we want to try and maintain that central vision through this process to either slow or stop the degeneration of your central vision, which may really help keep some quality of life. And as you know, retinitis pigmentosa is a relatively slow moving disease. So even if we were able to slow it down by a good percent, that you could possibly maintain some standard of life that you're used to right now and prevent any further deterioration, or at least slow that rate down. So that's one of the ideas that we had for this whole idea of gene-agnostic approaches. And this is nothing to say anything negative about those who are targeting one gene. That's a great way of doing it, that you go after one gene. And obviously with my extensive background with Luxturna, that has also been a great way to go. But we are looking for a way that maybe we can treat a larger group, and there are some of those genes that might be a little too rare for actual development. But we want to try and see if there was a way that we could develop this further. So that's one of the gene agnostic parts. And this is for retinitis pigmentosa, at least one of the rod cone dystrophies. There are other rod cone dystrophies that we can also consider as time moves on. This is in what we call the preclinical IND enabling phase, where we're doing studies in animal models and making sure this is safe in what we call NHPs, or non-human primates. So we're doing those things, trying to get enough data that we can go to the FDA and be given permission to go into a phase 1 or the first in human study. And if everything goes right, we're hoping that that will be sometime by the end of next year that maybe we'll be able to do this. Now, on top of this, we have a second thing, or a second program that we're also looking into. And this was developed by a researcher also at the Institut Television, Professor Denise Dalcara. And that uses a slightly different gene. And what happens is that as the disease, or as retinitis pigmentosa continues, those central cells, the ones that are responsible for light and daytime vision and acuity, color, things like that, they'll lose something called the outer segment, which is like a little projection that comes off the cell body. And in that projection, a certain process happens. It's called phototransduction. And phototransduction, really what it does is it takes the light that comes into the eye and goes through this process that converts it into an electrical impulse. And that impulse is sent through the retina to the brain, and the brain then deciphers that as vision. So without that kind of appendage, or that little arm that sticks out from the cell, that outer segment, that does not occur. But in her research, she found that using a certain gene called Xlrp, which is a G protein inward rectifying potassium channel gene, that if you put that in the cell without that outer segment, that arm that sticks out, you're able to do a shorter form of that phototransduction. So now you've actually restored that cell and enabled that cell to send that electrical impulse to the brain again that wasn't there before. And with that, in animal models, they've shown that there is a regaining of that cone function, or that central vision function through different mouse models. Now, I understand that mice are not little humans, but this gives us a little bit of indication that maybe there is some therapeutic benefit that we can do. And this is what we usually do when we get data and we give it to the FDA, and then the FDA will let us know if we can proceed. So that's another gene-agnostic way of doing things, because that, again, it doesn't matter why and how that outer little segment went away. It's simply that now we can actually go into that cell body itself and replenish and try to restore. So one of our tactics is to slow or stop the progression, and another one is to actually restore function. Now, if both of these work, and we're talking obviously a few years down the road, we may think of putting those together. So you'll actually have a function where we've restored some function, and then we've actually slowed that process down of that effect going away. So that is one of those things that we are also looking at. But right now we're concentrating on the first target, which is all about the RDCVF. We call that SPVN06, and that is our main target right now. That is our priority. Now, in the meantime, we have done a lot of work because we also wanted to go into the gene editing field. Because as I mentioned, we're a genetics ocular company. And so we didn't want to do just gene therapy. So one of those things about gene editing, you're going to hear more about that from the other speakers. So I'm not going to get into a lot of the details of that. But there was a company known as Intelia Therapeutics, and they were one of the first ones to show that they could actually edit the human genome in a systemic disease recently this past year. And we were fortunate enough to build a collaboration with them to explore ocular targets using this gene editing technology that they have. And so this is brand new. We just announced this about a month ago. So things are a little new for us, but we are rapidly looking into how we can move this all forward and in partnering with them. So we're very excited about this collaboration. So if I didn't mention before, we are a Paris-based company. So it means a lot of trips to Paris. I know that I can't complain about going to Paris. Nobody seems to have sympathy on me about that, but I do travel there quite a bit, and it's a great pleasure. But we're also going to be starting a center in Philadelphia, in the West Philly area, and we're looking for space there, and hopefully we'll be finalizing the US headquarters space there as well. So all that to say is that we are looking at these different targets. Some are gene agnostic, some are gene editing, and we really hope that we can start sharing a little more about this and some of our data as we move forward. And again, just a pleasure to be with you, and I hope to be taking some questions at the end, and I will throw it back to you. I think I'll give you back a few minutes here.
Okay. Thanks, Dan. That was a really compelling talk about your cone-targeted therapies. I love the fact that you're not only trying to save cones, but you have an approach that may be able to restore cones for vision. And the real home run would be to do both with a single therapy. And I also wanted to mention that the Foundation Fighting Blindness has what we call our Venture Philanthropy Fund, where we are investing and sometimes co-investing in companies to help them get into early stage clinical trials. And one of our investments is SparingVision, and we're really excited about the progress that's been made. It's been a long time coming. I know the work on this effort extends at least 15 or so years, probably closer to 20. And now we're getting close to a clinical trial. So that's really, really exciting. And again, thanks, Dan, for giving us that really clear and compelling overview of what SparingVision is doing.
And we appreciate FFB's support on that. So we look forward to a very long-standing relationship with the organization.
Well, it's definitely our pleasure. Thanks, Dan. Okay, so now we're moving on to our next speaker, and he's got the perfect first name for trying to address blinding retinal diseases. Bright Ashimatey. Dr. Ashimatey is a clinical research scientist at AGTC, where he's supporting the development of innovative study designs and endpoints for their gene therapy trials. And those are an Xlrp and achromatopsia. He's a vision scientist with background training in clinical optometry. He received research training in multimodal retinal imaging and functional assessment at the Indiana University School of Optometry, and he pursued postdoctoral training in ophthalmology at the USC Roski Eye Institute and Johns Hopkins Wilmer Eye Institute. So Bright, the floor is all yours. You can share your slides and begin.
Thank you, Ben, for having me. I really appreciate the opportunity we have with this platform to share with your audience the work we are doing at AGTC. Can everybody see my slides? Then do you see my slides?
We can over here, yes.
Okay.
They look good, thank you.
Thank you. And so I would like to start by thanking your audience today who continue to journey with us on our journey to fighting blindness. AGTC is a publicly traded company, and so we have a forward-looking statement. So picking up from where Dr. Chung left off, we are focused on a number of disease conditions, which are inherited retinal disease conditions. And I would like to start off by telling you some results we are getting from our X-linked retinitis pigmentosa trial. So for some of the audience today, blindness remains a very devastating thing. And because they themselves or someone they know or a family member or someone they care for has this blinding condition. And we at AGTC take our commitment to investigating and developing treatment trials for inherited retinal disease conditions very seriously. And we are committed to doing so in a very safe manner. And so focusing on Xlrp or X-linked retinitis pigmentosa, as Dr. Chung mentioned earlier on, this is a disease condition which is caused by an abnormality to the gene responsible for producing some proteins which are really critical for the functioning of some cells in the eye we call the photoreceptor cells. And these are cells which transform light into electrical signals for the brain to interpret. And this condition affects approximately 20,000 patients in the US and in the EU. And currently there is no there's no treatment modality for the condition. Patients who have the condition will often start having issues with having seeing in low light levels. And this is often referred to as night blindness. And as the disease progresses, they start losing their vision from the periphery, and then eventually the vision in the central field is closed off. By the time those patients are age 45, they will be termed legally blind. And so our overarching goal in AGTC is to develop a gene therapy program which aims at delivering the highly optimized and working genes into the cells, the photoreceptor cells. That way we can improve their function and, if nothing, be able to preserve the function that they are at the time of the gene therapy. And so to be able to do this, we have multiple ways of assessing the way the subject's vision is before treatment, as well as assessing those functions after treatment. That way we can be able to investigate the efficacy of our treatment modality. And some of the techniques we use include visual acuity testing. And this is a very common form of testing in the clinic where the patient is asked to close one eye and read characters on a chart, and then is asked to close the other eye and then read characters on the chart. That way the doctor or the clinician is able to tell how crisp the patient's visions are. Another form of testing modality is what we call the visual field sensitivity assessment, or the MAIA. This is also a very simple testing modality which doesn't hurt at all. it presents light to the patient, and the patient's goal would be to respond when they see the light. And so the lightening conditions will be varied from a very bright light to very low dim level lights. And the goal of the clinician is to assess the dimmest possible light that the patient is able to see. Both visual acuity testing and microperimetry or visual field testing are very useful ways of providing the information the clinician or the doctor needs to be able to understand the way the patient's vision is as of the time they are presenting in the clinic. We at AGTC are very focused on understanding the experiences of the patient. And so we are beginning to introduce the orientation and mobility maze into our testing protocols. And this is going to help us directly visualize or understand how our treatment modality is impacting the livelihood of the patients. Because for most of these patients, what they want to be able to do is to be able to move around and see clearly. And so by introducing the orientation course, we will be better able to understand how our treatment modality is impacting the livelihood of the patient, because again, the patient is our point of focus. And so we have been conducting some clinical trials, and clinical trial is the way of investigating the safety as well as the efficacy of your treatment modality in a patient population or in a human population. And the first stages of the trial is aimed at assessing the safety of your drug development or of the drug. And then the second part is focused on understanding how effective your drug is working, as well as seeing again the safety profile of your drug. We have completed the first stage of our clinical trial, and so far we found that our drug, the AGTC 501, is very safe. The inflammations remain well controlled, and there is no evidence of retinal structural damage post-treatment. And all the patients that we have dosed so far, all the patients who participated in our clinical trial so far, continue to demonstrate very favorable safety profiles. With regards to how well our drug is working, we are finding very encouraging and durable improvements in the visual acuity of the patients who have participated in our trial at month 4. We are also finding that the visual field assessments that we have performed in the subject, or the improvements in visual field assessment that we have found post-treatment of the patients who participated in our trial, continue to sustain even at month 12 for the few subjects, at month 24 for the few subjects for the few patients who have reached 2 years of our trial. And so we are very excited about the findings so far from the patients who have participated in our trial. And so we are continuing to investigate the safety as well as the efficacy profile of the patients who will be enrolling in our future trials. So if you are interested in participating in our trial, what should you expect? So when you enroll on our trial, we usually would follow you for 5 years. That is, you will have clinic visits a number of times in the first year, and then once every year until year 5 when the trial is completed. The reason for the many visits in the first year is because we will be interested in assessing your vision at baseline, and then we will administer our treatment. And then post-treatment, we are going to follow you to make sure that the medications are safe. And then we will start assessing the efficacy of the treatment modality. As in, we'll start checking your visual acuity and then your visual field sensitivity, as well as now your ability to navigate a mobility course. And that would help us quantify the improvements that you have gained due to the treatment administered from our drug. And so I would like to now talk about the 2 trials which we have going on in Xlrp at this point. One of them is Skyline, and this is somewhat in the middle between the first steps of the trial as well as the second steps of the trial. And this trial will be dosing patients on two doses of the current regimen that we used in the early stages of the trial. And the goal again over there is to assess the safety of those 2 doses, as well as the tolerability of the 2 doses. And again, we will be asking the subjects to walk through a maze course, and that is going to help us better quantify the effects of the treatment modality on the livelihood of the patient. Then we will be moving on to the next stage, which is the Vista Study. And the goal of this study would be the primary goal would be to assess the efficacy or the improvements that the subject is gaining, that the patient is gaining post-treatment. And currently we are screening subjects for this study. And if this is something you would be interested in, or somebody who would be interested in, please go to www.cynictrials.com to find more information about that. Or you can send an email to advocacy@agtc.com and someone would promptly respond to your email. So then, having talked about our Xlrp program, I want to also spend some time to talk about a few other disease indications that we have been investigating. One of them is achromatopsia. And achromatopsia is a condition where the patient is unable to, or the patient feels very uncomfortable under normal daylight when anybody with functional vision should be okay. This patient would also often have very reduced visual acuity. We have completed the first steps of the clinical trial process, and we have found that the treatments administered to these patients are very safe, and we are also finding some really encouraging results in the patients with the B3 gene mutation. And if you are interested in knowing more about that, I invite you to visit our website to read more on that as well. Apart from focusing on inherited retinal disease conditions, which affect primarily which we start seeing the effect from young people until middle age, we are also developing products in dry age-related macular degeneration, which is the primary cause of visual abnormality in the aged population. And we are taking advantage of some novel pathways which we have identified to be able to at least halt the progression of the disease course. And so we are covering both the young population as well as people in the older population. Apart from our own trials, we have also been partnering with others, with our peers in the space. Dr. Chung just spoke about the very wonderful work going on at SparingVision. And we at AGTC are proud to collaborate with SparingVision by licensing our cone promoter to the program. We are also partnering with Bionic Sight on their optogenetics program. And optogenetics is designed for people who have lost their photoreceptors, or the cells which are responsible for converting light into electrical signals for the brain. In subjects who have completely lost those, there is now the ability to put in photosensitive proteins into their eyes, and those photosensitive proteins are able to help the patient perceive light. So far, Bionic Sight has those 4 subjects, and 4 subjects who are blind or close to being blind. 2 of them are now able to, all 4 of them are now able to perceive light, and 2 of them are actually able to perceive the direction of the light. We do also have some collaboration with Team on the Xlrs, or X-linked retinoschisis program. So Ben, I would like to end by saying that we at AGTC feel very excited about the advances we have made with drug development programs and the promises they hold. And we are thankful that we are making this journey together with our peers, our patients, their families, and their caregivers. And we look forward to celebrating the successes that we can achieve together in winning the fight against blindness. I will pause here, and then I will see you again during the Q&A section. Thank you.
Thank you, Bright, for that really excellent presentation and overview of your Xlrp and achromatopsia gene therapy trials. Really encouraging work and a really well done presentation. I wanted to comment on a couple of points that you made that I think are important for everybody, even if they don't have X-linked RP or achromatopsia. And you talked about the number of visits that one has to make when they're in a clinical trial. And I think it's important for people to understand that when people sign up for a clinical trial, it really is a big commitment because there are a lot of visits, there's a lot of testing. You can't just pop in one day and be done. It's a very long-term commitment. And another point that you touched on, which I think is very important, we always get a lot of questions about why these clinical trials can take so long. And of course, you want to see that safety is maintained over the long term. You want to check for many years for safety, but you also want to ensure that there's durability of the therapy. If it works after 1 month, that's great, but you want to see if it can work after a year or 2 years or even longer, because that's important if the therapy is going to succeed. So I'm really glad you touched on those points, and I just wanted to emphasize those.
There is one thing I want to add quickly to your point, Ben, is that we are also paying for the patients and as well as their caregivers when they participate in our trials. And so we pay for their transportation, we pay for their lodging in, we pay for all the materials that are used for the study and so on. And so we understand that we are making this journey together with the patients. And so we are committed to making sure that we are taking away every possible obstacles that can hold them back from participating in our trials. And so that is just one other thing I want to put out there. In addition to all that we do, is we also are very interested in making sure that when you want to participate in our trials, nothing actually holds you back financially. Thank you.
That's a great point to make. And I'm sure everybody appreciates that financial support, because that is not trivial. So thank you for saying that. Okay, and if you can stop sharing your slides so we can move on to our next speaker. And our next speaker is none other than Claire Gelfman, the Foundation's Chief Scientific Officer. And I have to say, Claire, working with you has been a real pleasure. You've picked up on our space so quickly and so deeply. It's really impressive. I know you've only been, I think you've been with the Foundation now, is it 6-7 months? It seems like several years with your knowledge and your enthusiasm. So thank you for all you're doing to advance our mission and help patients and families, and obviously presenting today. So now I am going to read your formal bio so people can know a little more background on you. So Dr. Gelfman joined the Foundation Fighting Blindness as Chief Scientific Officer in April 2021. I was right, it's about 6 or 7 months. She is responsible for leading the overarching scientific strategy for the organization, with a focus on research initiatives designed to accelerate new treatments and cures for inherited retinal diseases. Additionally, she oversees the Foundation's non-clinical research portfolio, including early translational and preclinical research programs. She was previously Vice President, Pharmaceutical Development at Adverum Biotechnologies, an ocular gene therapy company developing AAV-based treatments for wet age-related macular degeneration and inherited retinal diseases. So the floor is yours, Claire.
All right, Ben, thank you so much for that introduction. Let me share my screen. All right, let me just make sure.
Looks good.
You guys can see it. All right. Well, thank you so much, Ben, for that very, very nice introduction. It's a pleasure to be here. And, you know, you mentioned earlier that we are celebrating 50 years around our mission. It's a long period of time, but I'm here to tell you that our urgent mission, we have not wavered on that regarding driving the research that's going to provide preventions, treatments, and cures for the entire spectrum of retinal degenerative diseases. And I'm also happy to say that we're the world's leading private funder of efforts in this area. So let's get started then. What are the facts? So there's about over 300 genes now that have been identified that are associated with various inherited retinal diseases. And the most common of those is retinitis pigmentosa. It covers about 2 million people worldwide with an incidence of about 1 in 3,000. And there are about 70 known mutations, 70 known genes when mutated are responsible for that RP diagnosis. But there are a lot of other IRDs, and we've heard about some of them already today, including Stargardt's, choroideremia, Leber's congenital amaurosis, or LCA, achromatopsia, and also retinitis pigmentosa associated with Usher syndrome. So because all of these are what we call monogenic, and that a single gene is responsible for compromised vision, it's no surprise then that one of the approaches that we've heard most about up to now has been what we call gene augmentation therapy, where you bring in essentially a good copy of the gene to correct the defect. And this is really all about knowing the fact that when we start with a molecule of DNA, that molecule is then transcribed to a molecule of RNA, and that RNA then is translated to make a specific protein. And that process is really important because it's the proteins in every cell in our bodies that have to be present and functioning properly for our cells to function properly. And so when we think about more traditional gene therapy with the augmentation of a gene, we think about a special vehicle that will deliver that correct gene. And that special vehicle is an adeno-associated virus. And I think one of the ways I think about explaining this is think of this vehicle as a bus, and the bus is full of children that need to be taken to the correct school. And so the outer shell of the bus is kind of like this protein coat, if you will, the vehicle that's going to be driving the correct gene, or the children in this example, to the correct school. So without the correct captured, or the correct bus, if you will, to drive the correct gene to the right place, that's what gene augmentation is all about. We're providing a functional copy of that DNA so the correct protein can then be made. So the best example of that, as was already mentioned, is Luxturna from Stargardt Therapeutics that was approved in 2017 that really reignited the gene therapy world. And it's the first AAV-based gene therapy to be approved for an inherited retinal disease, specifically Leber congenital amaurosis, or LCA type 2, which is caused by mutations in a gene known as RPE65. So no surprise then that this gene augmentation therapy is whereby a subretinal injection of this gene therapy is delivered under the retina to make a functional copy, to deliver a functional copy of the DNA so that the right protein, so the RPE65, can be made in the right place. Well, another type of gene therapy that we've heard a lot about lately is gene editing. It's been all over the news, not only in the ophthalmology field, but CRISPR, you also hear a lot about that outside the ocular field. And there's a company called Editas, and I think Ben's going to focus on this in more detail later, but they have a gene editing program for a different type of LCA known as LCA10. And this company is in a phase 1/2 clinical trial for individuals with LCA10 who have mutations in a gene known as CEP290. And recent results show not only safety in these individuals, but early efficacy in 3 subjects in the mid-dose after about 1 year. So we'll look forward to hearing a lot more about that as well. Well, these gene augmentation and even gene editing approaches are great, but what if your gene has yet to be identified? Or what if your specific gene has been identified, but its development happens to be sparse? Because these are all rare diseases that we're talking about. So I want to introduce this concept of mutation agnostic approaches, where it's a way to think about treating inherited retinal diseases that's not necessarily dependent upon knowing your specific mutation. And these mutation agnostic approaches would result in treating more IRD affected individuals with a single therapeutic. So what do I mean by that? This is going to be based on the pathology of the disease. So these mutation agnostic approaches are really based on the pathology, knowing what happens at the cellular level. Well, we already heard a moment ago about the importance of the ability of rods and cones in our photoreceptors to trap photons of light that are coming in and then engulf that, if you will, those photons of light. And then a message, this electrical impulse, is sent to our brain via our optic nerve. And our brain can actually then interpret through that signal what image is in front of us. And that's a very important part of proper vision. And when that's compromised, when our rods and cones cannot carry that out, that's when vision is compromised. And so I'm going to go through a couple different examples of these mutation agnostic approaches, a couple examples that are actually gene therapy based, but not based on knowing an individual's mutation, but rather using gene therapy to help a cellular function to get at that pathology that we were describing. And one of those we've already heard about from Dan Chung from SparingVision. It's really more of a neuroprotection with this rod-derived cone viability factor. And then we'll also talk about some non-gene therapy-based approaches that are also mutation agnostic. All right, so we'll start with optogenetics. And so optogenetics is one of these techniques that involves the transfer of a gene that encodes for one of these light-sensing proteins. But what's important about that is that it can transform a cell that normally is not light-sensing into one that can now respond to light. Because with optogenetics, we're looking to treat individuals who've really lost all function of their photoreceptors. So we can't rely on a functional photoreceptor, and optogenetics does not have to have that, which is wonderful. So what we're essentially doing here is we're giving that light sensing ability to other cells in the retina, to our interretinal cell bodies, as well as ganglion cells. And that's not only being leveraged by, for example, bionic sight that we just heard about, but also GenSight I wanted to mention. I was actually just at a conference last weekend, the American Academy of Ophthalmology, and we heard some very new data that came out of the GenSight trial that is a type of gene therapy where a special protein is being given to allow for that, that light sensing ability to be restored to cells that normally don't have that function. And it was the second individual in this trial who, in this case, started with no light perception, that in the presence of wearing these special goggles to concentrate the light coming in, specific for this type of therapy, was able to, for the first time, actually identify some pens on a table that were spread out. And this was actually the second participant in the study. So we're looking forward to getting a lot more data coming out as more of the individuals will be reading out those results. And there are other companies in the space that are really leveraging optogenetics as a way to treat retinitis pigmentosa in a mutation agnostic way. And I've listed a couple of those companies here, including both Nanoscope and Videri Bio. Well, I've got on this next slide some other mutation agnostic approaches that again are based on not so much an individual's causative mutation, but what we know about the rods and cones and their degenerative process. So one thing we know is that oxidative stress plays a pivotal role in developing and also accelerating retinal degenerative diseases due to the buildup of these free radicals that are toxic. And oxidative stress is part of the pathology, not just for ocular diseases, but really, you hear about free radical generation in the cancer world. And what happens is, our bodies are constantly undergoing oxidative stress. But thankfully, we have these inherent cellular mechanisms to protect against it. But as we get older, those mechanisms aren't quite as strong as they were when we were younger. And in the eye, for example, the high oxidative nature of the retina really accelerates that process. So knowing that, it's no surprise then that the therapeutic strategy for targeting oxidative stress may be important for the treatment of these inherited retinal diseases. And so there's a company in the Fort Worth area called Acuity that has a product in phase 1/2, and they're using a small molecule, and that small molecule is called N-acetylcysteine amide, also known as NACA, and it's being evaluated in a clinical trial in Australia. It's in a population of Usher patients, but it's specifically looking to treat their retinitis pigmentosa. So I mentioned before that oxidative stress is really deleterious to cells. And what this therapy is doing is it increases cellular glutathione levels. And it's these glutathione is an antioxidant. So it's a way to bring in a smaller molecule, given systemically, by the way, not directly into the eye, but given to the body, that increases the levels of an antioxidant to then protect against this oxidative stress that's so deleterious to photoreceptors. So another mutation agnostic approach has to do with modulation of another pathway called complement. So complement is really important in our bodies as well, because it helps protect against pathogens. And so what can happen in a lot of different disease states is the complement pathway just goes on overdrive. It goes haywire, and no exception in the ocular space as well. And so a lot of treatment modalities that are affecting the complement pathway are all about inhibiting it, but not inhibiting it completely, because we still need it to be active, but not overactive. And so Iveric Bio has a product in phase 2 through their STAR trial. This is actually for Stargardts, but it's a mutation agnostic approach whereby the intravitreal delivery of this compound is inhibiting a pivotal inflection point along this pathway that can inhibit the deleterious effects of inflammation and cell death. And this whole complement modulation, you'll hear a lot about that too for dry AMD. There's some studies that are going to be reading out soon for potential treatments in that area as well. So, I've shared with you some gene augmentation therapies, also gene-specific gene editing through CRISPR. One of the things I didn't mention was RNA-based therapies, whereby an antisense oligonucleotide can also be used in order to either turn on or turn off a specific gene, depending upon what therapeutic direction it needs to go. But I see these gene-specific therapies as part of our toolbox. They're really important, but we want to have multiple shots on goal here. And so now we've got these mutation agnostic approaches, and I mentioned just a few of those, and hopefully we're going to add even more things to that toolbox. And I didn't have time to mention today that there's also cell-based therapies that are in development, as well as retinal prosthetics that are in development. So the more we can do to add to this toolbox will give us a better chance of finding treatments for both inherited retinal diseases as well as the dry form of age-related macular degeneration, what we have been working so hard as a foundation for decades on. So in conclusion, these mutation agnostic approaches really complement that toolbox, and because they could treat more inherited retinal disease affected individuals with a single therapeutic that will allow for targeting pathways of photoreceptor degenerations that are common to all IRDs. And this is really all about coming up with more shots on goal to prevent and come up with treatments and cures for individuals suffering with IRDs. So I will stop there and be happy to answer any questions when we get to the Q&A. Thank you for your attention.
Great. Thank you, Claire. That was a really great presentation, and thanks for underscoring the promise of gene-agnostic therapies for helping a very large swath of people affected by inherited retinal diseases. One of the big challenges in our space is there are somewhere in the neighborhood of, depending on who you talk to, almost 300 genes, each of which, when mutated, can cause a retinal disease. And that makes developing therapies really challenging. But with these gene-agnostic approaches, hopefully you can help bigger groups of people by not worrying specifically about their gene. So thanks again. And our next presenter is yours truly. Let me bring up my slides here. Can people see that? Oh, can everyone see that okay? Okay. I see nods, so that's a good thing. And let me get it into presentation mode. So to close things out here for the next 15 minutes or so, I wanted to cover some of the newer announcements and advancements in our space. And also my final slide covers Stargardt disease, because that's something that is, it's actually the most common retinal degeneration from a genetic standpoint, ABCA4, and we always have a lot of people tuned in who are interested in what's happening with Stargardt disease. So first, this is a good segue from Claire's talk about the complement system. But first, I want to talk just briefly about age-related macular degeneration. So unlike the inherited conditions we've been hearing a lot about today, AMD is the most common retinal degeneration. It's the most common form of blindness in people 55 years and older. And it's a complex condition. Yes, there are genetic factors, but unfortunately, aging is a big factor. If you smoked, that's a big factor. And there are other potential lifestyle factors. But one key pathway that researchers have identified in AMD is the complement system that Claire was talking about. It's that overactive immune system that causes damage. And in people with AMD, they virtually always start off with what we call the dry form. And the dry form is this accumulation of deposits underneath the retina, and those ultimately cause the damage, but the complement system seems to be driving that. So the company Apellis has been developing a complement inhibitor. It's targeting a specific pathway called C3, a specific protein, with a treatment, an emerging treatment they call pegcetacoplan. And this treatment in the clinical trials has been injected every month or two into the middle of the eye, into the vitreous. And so a couple of months ago, Apellis reported that in their phase 3 clinical trials at 12 months, that they had some encouraging results. And in the combined trials, they've had two trials underway, Oaks and Derby. The trials reduced the growth of lesions that are caused by advanced AMD by 17%. Now, what happens in advanced AMD when it gets to the point where it's causing degeneration of the cells, we call that geographic atrophy. And geographic atrophy is characterized by these lesions. So this trial was reducing the lesion growth. Very encouraging. Now, they had two trials. One met its endpoint. The other just missed. But collectively, if you put all the people together in these two trials, again, lesion growth was reduced by 17%. So Apellis is seeking FDA approval for their medication. They plan to in 2022, so stay tuned. What's important is currently we have no dry AMD treatments, no treatments for GA. So it'd be really great if we could get something across the finish line. So now I'm going to move to wet AMD. So I said just in the last slide that all AMD usually starts off as the dry form, the accumulation of these deposits underneath the retina that can themselves do damage. But what happens in a smaller subset of people is they develop this secondary phenomenon called wet AMD, or technically it's called choroidal neovascularization. But the growth of these leaky blood vessels underneath the retina can do a lot of damage, and it can happen more quickly and more severely in many cases than dry AMD. Now, the good news is for many years, we've had many treatments FDA approved to address wet AMD. Things like Lucentis, Eylea, some physicians prescribe Avastin, a cancer drug, off-label, and then the newest, I believe, is Beovu. Now, the challenge with all these approaches, they do generally work pretty well, but they involve regular injections into the eyes of patients, really for the rest of the patient's life. These are, again, intravitreal injections into the middle of the eye. And that basically means the patient has to go to their eye doctor on a regular basis to get these injections. It could be as often as monthly. But the good news is, just about a month ago, Genentech announced FDA approval for their treatment approach for wet AMD called Susvimo. And what this is, it's a tiny capsule or a tiny reservoir. They actually call it the port delivery system. It's only the size of a rice grain, and it's implanted just under the surface of the outer part of the eye, just underneath the sclera, or right at the edge of the sclera. And instead of injecting a treatment into the eye, this capsule, or this port delivery system, is refilled. And what they found in their phase 3 trial is it only needed to be refilled about every 6 months. So instead of getting regular injections into the eye with this port delivery system, you only need to get it refilled perhaps every 6 months. And in fact, Genentech is hoping in additional studies and analyses to see if they can extend that even further. And what they're putting in this capsule is somewhat similar to their drug Lucentis. It is different, it's similar but different. But what's cool is it provides sustained release to inhibit the growth of those leaky blood vessels. So this is a big step forward. It's not available yet. Hopefully in the next several weeks or couple months it'll be available. Not all eye doctors will be able to implant the device, and it may not be suitable for all patients, but for many people it's an excellent alternative to reduce that treatment burden of those regular injections into the eye. Okay, so let's switch gears, move back into the inherited retinal disease space, and I'm really excited to talk about a new company launched by the Foundation Fighting Blindness. And you've heard me mention a time or two during this webinar about our venture philanthropy fund, the RD Fund. Now that's an investment fund where we are investing in, many times co-investing in, companies that are developing therapies that are at the doorstep of a clinical trial or maybe in an early stage clinical trial. This fund is really geared toward boosting clinical development for promising therapies and get more into the clinical trial pipeline so we can get them across the finish line. We've actually invested in 11 companies, more than $100 million at this point. But what's exciting about Opus Genetics is it's the Foundation's own company. We've created our own company with the purpose of targeting the even rarer retinal diseases. These are conditions that have been not addressed by other companies. And our initial targets that we've announced are for LCA5, which is caused by mutations in the Leber-cillin gene, RDH12 or LCA13, which is caused by mutations in the Leber-cillin, LCA13, which is caused by mutations in the RDH12 gene, and LCA9, which is caused by mutations in NMNAT1. Again, these are rare conditions that, of course, all cause LCA and have not been previously addressed in the clinic. So this company has gotten off the ground with $19 million in initial seed funding from the Manning family, their foundation, and BIOS Partners. Our scientific co-founders include Gene Bennett, who's the visionary behind Luxturna, Junwei Sun of University of Pennsylvania, and also Eric Pierce of Mass Eye and Ear. So this is our company. We're taking on all the responsibilities of a company, hiring the staff, raising the capital, even we're responsible for manufacturing of the gene therapies. So very exciting development for us, and stay tuned as we move these candidates toward the clinic, and hopefully in the not too distant future announce more candidates for treatment. Now, moving on to my next slide, I wanted to just talk a little more about the treatment that Claire talked about in her presentation, and this is a CRISPR-Cas9 gene editing emerging treatment being developed by Editas. And as you heard, unlike gene therapy, where you're delivering a whole new gene to replace or augment the mutated gene, CRISPR-Cas9 is gene editing. You can think of it as like a pair of molecular scissors that's just coming in to cut out or correct just the mutation in the gene. So you're just targeting the mutated area. And in some cases, you may paste in a little piece of DNA as part of the corrective process. So Editas, several months ago, launched their clinical trial of their CRISPR-Cas9 approach for a specific mutation in CEP290, which causes LCA type 10. And as you heard, there have been encouraging vision improvements for a couple of patients. They recently announced those. And I want to plug my Eye on the Cure podcast, which the new episode launched yesterday. And I really had the privilege and delight to interview a patient from that trial, this gentleman, Michael Kalberer of Long Island, New York, and he talked about what it was like to have vision improvements. He can see his food better. He can be in a restaurant and enjoy that experience better because he can see more in dim conditions. And he said his biggest complaint about receiving this therapy, he's afraid he's going to gain weight because he's enjoying food that much more because he can see better. So I think that's a pretty cool anecdote. So one interesting fact about this approach, it's the first CRISPR-Cas9 treatment ever to be administered directly to the human body. And it's very cool that the retina was the first target for this approach. The retina is actually a great target for treatments because it's relatively accessible. It's obviously in the front of our bodies. You can get to it relatively easily. It's a small target, and we have two of them. So you can treat one and compare how the treated retina does to the untreated retina. That's really good for scientists. So we're looking forward to hearing further results from this trial. And Editas is also in preclinical development for USH2A, specifically mutations that occur in the exon 13 region, and also they're targeting the rhodopsin gene, which when mutated is a pretty common cause of RP. So stay tuned to hear more about preclinical development of those. And I will add, I don't have time to get into it, but the Foundation Fighting Blindness is funding many CRISPR-Cas9 trials. And so finally, to close things out, I wanted to talk about emerging therapies for Stargardt disease. And I was watching the Q&A, and as usual, we got a lot of great questions about Stargardt disease. And I think a key thing to know about Stargardt disease is it's a vitamin A processing problem. That's what really leads to the development of the condition. So we all need vitamin A in our diets or through supplementation for our retinas to be light sensitive. Vitamin A is that magic molecule that combines with other proteins in the retina to make our photoreceptors light sensitive. If you eat a normal diet, you get plenty of vitamin A, so that's not a problem for us. If you are in a situation where you are malnourished or you don't get a good supply of food, you could actually have vision loss from vitamin A deficiency, but that's not true for people in developed countries for the most part. But if you think of vitamin A as being like a fuel for your retina, it's metabolized or processed in the retina to make it effective and to enable it to actually work. But like a fuel, when you process it, it leads to these byproducts which are toxic and harmful. Just like when we put gas in a car, there's exhaust that you have to get out of the car to keep the car running properly. Well, in the retina, when you process vitamin A, there are these metabolites which need to be cleared. And the problem in people with Stargardt disease is these metabolites accumulate in the retina, and that's what ultimately causes the damage. And again, so Stargardt disease is a metabolic issue. It leads to the improper processing of these vitamin A metabolites. So there are several companies that have targeted this metabolism problem. One of those is the company Alkeus, which has developed a form of vitamin A called deuterated vitamin A that metabolizes more cleanly than standard vitamin A. So in the clinical trials, they give people this vitamin A. The people try to limit their dietary vitamin A, and the hope is that that will reduce the accumulation of these byproducts. So informally, in their phase 2 trial, they have talked about some encouraging results. We're waiting to see more formal publication of those results and to see what their plans are for a phase 3. But that's some promising work being done by Alkeus. Now, the company Acucela has a drug that slows down the uptake of vitamin A to the retina as a way of hopefully reducing the accumulation of these metabolites or these byproducts. They're in a phase 3, and we're hoping to hear something from them in the not too distant future in terms of results from the phase 3. And then a company in our RD fund portfolio, which I talked about, called Stargazer, is in phase 2, and they're reducing the production of a protein called retinal binding protein 4 that is responsible for the uptake of vitamin A into the retina. And by doing so, we're hoping again to reduce the accumulation of those metabolites. Now, as you heard from Claire in the last presentation, the company Iveric Bio is targeting the complement system to address Stargardt disease. Researchers believe the complement system is overactive in people with Stargardt disease. Their approach is in a phase 2. We look forward to results from that, hearing about that. And then I just learned yesterday from one of my colleagues that the National Eye Institute has launched a clinical trial of a drug called metformin for improving retinal metabolism in people with Stargardt disease. And I apologize for the leaf blower outside my window here. I don't know if you can hear that. But anyway, metformin is an FDA-approved drug that's used for diabetes and other conditions. And it improves a process called glycolysis, which is a metabolic process in the retina. And I believe that's what the NEI is hoping they observe in patients with Stargardt disease. By improving retinal metabolism, you can improve the toxin accumulation problem. So they're just moving into a phase 1/2 trial. And then finally, I wanted to mention that there are several gene therapies in preclinical development for Stargardt disease. Most cases of Stargardt disease are caused by mutations in the gene ABCA4. The challenge with that gene, it's a big gene. It's hard to get it in the viral containers that are used to deliver healthy genes into the retina. So you really have to do one of two things. You have to shrink the gene so it fits in the container, or you have to deliver it in 2 containers. And so 3 companies are addressing this issue: AGTC, another company called Cove, and Iveric Bio. So stay tuned as these move through preclinical development and hopefully before long in clinical trials. So before I conclude, I want to go back to my opening introduction where I mentioned you can get a lot of great information on just about everything you heard today and so much more at fightingblindness.org. If you're interested in learning about clinical trials, what the criteria for being in the trial is, what sites the trials are being held at, and how to reach out to the trial coordinators to let them know you're interested, you can go to clinicaltrials.gov. And then another great way to be on the radar screen of therapy developers, companies, and researchers is to register in our My Retina Tracker registry at myretinatracker.org. By putting information in there, if you have genetic information, that's great. If you don't, that's okay as well. But companies and researchers look at profiles. They never get your personal information, but they look at profiles to identify potential participants in their clinical trials. When they do those searches, the personal information is available to foundation administrators. We contact you and let you know you match the criteria. Then it's up to you as the patient or the family member to contact the trial sponsor. So a great thing to be in the My Retina Tracker registry. So that concludes our talks. I am going to stop sharing my screen. And we have about a half hour for talks. And for questions and answers. And the first question I wanted to pose to Dan Chung, because this is actually a question I get frequently outside of the webinars. And so for the SparingVision treatment, you're targeting cones. And I know you're targeting at first RP patients and potentially Usher syndrome patients. But what about people with other conditions where cones are affected, like Stargardt disease or Best disease or cone rod dystrophies? Is that on the SparingVision radar screen?
So it definitely is on the radar screen. I think at this point we're looking at other preclinical studies to really determine the safety of that. One of the things that we look at when we think about the rod-derived cone viability factors, that you have the loss of your rod photoreceptors and the loss of the production of RDCVF that is supporting the cones. When you look at cone rod dystrophies or cone dystrophies, you still have a relatively good complement of rod photoreceptors. So the idea is that if we were to overexpress this with the idea that we would help to save cones, really what is the effect that would have on the other cells? So those are definitely questions that we're entertaining. The idea right now is obviously we want to concentrate on developing this and getting this into the clinic, the RDCVF, but those thoughts are definitely in the conversation right now, because in the end, we do want this therapeutic to be gene agnostic, in the sense that if we can see safety and efficacy in preclinical models of other retinal diseases, then we will definitely look into their clinical development.
Great. Well, that's encouraging to hear. So thanks for mentioning that at least those other conditions are on your radar screen. You're thinking about those other conditions. Another question we got about the SparingVision treatment approach, the one to save cones, RDCVF. How many applications do you think a patient will need, or what's the hope? How many injections?
So the idea is this would follow the standard of what gene therapy is, and that is it's a one-time treatment in the sense that, especially with AAVs, they're non-integrating viruses in the recombinant form, and they form a little structure called an episome, and that from there, it produces the protein that you're trying to get, and obviously in whatever target cell you're going for. So the idea is one injection. I know in the field there has been talk about re-injection and things of that nature, but safety studies would have to really be done to really further that approach at this point. So the idea right now is to have that one injection as a gene therapy product.
Okay, great, great. That would be great if you would only need one injection for your lifetime.
Absolutely.
Thank you. So, Claire, I have a couple good questions, and they're frequently asked questions, but I think these are important to address. So, for those of us that have been paying attention to the COVID vaccines, we've learned that the Pfizer and the Moderna vaccines use mRNA to be effective. And there are also RNA approaches to treating retinal disease. Can you talk about any commonalities, or probably more importantly, the differences between those two?
Sure. No, thanks, Ben. So, right. So the mRNA vaccines, obviously a lot of attention. And it's when we think about sort of genetic ways of treating, not only IRDs, but also, things like COVID, gene therapy is being used. It is a gene therapy based treatment at the level of the RNA. It's an mRNA vaccine, and it's making the spike protein that we've heard about that's important for infection. And so while that is a type of gene therapy, it's different in the RNA-based gene therapies that are being used to treat inherited retinal disease, because those are more specific for the genes that we know are mutated, that have been identified as being mutated. And in some cases, we need to restore function of a gene that's inherent to the photoreceptor cells. Other times, we need to inhibit that. And so some of these RNA-based therapies are doing just that. So it's a little bit different in those specific ways.
Okay. Good answer. Good answer. Another question for you, Claire, and I think this extends beyond just the gene-agnostic therapies that you talked about, but if you participate in a clinical trial for any therapy, maybe a gene therapy, a stem cell therapy, is that going to preclude you from being in another trial down the road?
Yeah, no, it's a great question. And with a lot of these gene-based therapies, like the one that Dan was mentioning, if the idea is that you're going to get it one time, that means that if it's working, it's continually expressing the therapeutic protein, hopefully, forever. That's really the idea there. If you already have that happening, usually if a company will put in their, exclusion criteria, so the things that, that are okay, that you need to have when you enter a trial versus the things that you cannot have when you enter a trial. Most of the criteria that I've seen, if you've been in one trial, then unfortunately for gene therapy, you would not be able to enter another. Those decisions, though, are made by the sponsoring company, and those decisions are made with great discussion with key opinion leaders, with physicians, with the regulators, to make sure that those decisions are made so that there's no safety issues. Obviously, safety is the number one thing to consider for these clinical trials, and if there's any possibility that getting something, if you've already been in a trial, then it won't be allowed. So it's specific, for the individual trial, but particularly for gene-based therapies, unlikely.
Right. Thanks for clarifying that. Okay, so I have a good question for Bright. Bright, we had somebody ask about your Xlrp trial and whether a second eye, I guess the first question is, in either your Scenic trial or your Vista trial, will you be hopefully treating second eyes? Is that part of the protocol? And the specific question we got is, does the second eye require a different vector?
Hi, Ben. So the goal would be to treat both eyes ultimately. And we have discussed that at this point, our goal would be to gather the data in one eye, and then we'll be having a meeting with the FDA and discuss the data, and then ask inputs from the FDA. And once the FDA gives us a go ahead, we will be targeting those in the second eye too. Will the same vector be used? The answer is yes. And it's because if we administer treatment in one eye and the vector is working fine, then we just have all the confidence to go ahead and dose the second eye with the same vector. And so that would be the goal.
Okay, great. That's excellent to hear. Thank you for that. And the next question I think is a really good one, and I want to open it up to all the panelists. It's about the use of the phrase shots on goal. And I'm just reading the question here. And I guess I'm going to look at this question a little differently than the way it's asked. We often talk about shots on goal in clinical development, that you need a lot of shots on goal until you get a therapy that's approved by the FDA or another regulatory body. And I was wondering if one of our panelists wants to talk about why many shots on goal are needed and just kind of make a general commentary about clinical trials.
I guess I'm happy to start on that.
Okay.
I think that for the community in general, the more different modalities you have to tackle a singular problem is definitely heightening your chance that something will work in the end. In every clinical trial, there's a lot of research that is done before you actually get to the point where you're testing products in humans. And with that, you never quite know how clinical development is going to go. Animal models and in vitro models and things give you a good idea of the safety and give you ideas that there's a possibility that this will work in human trials. In human trials, it's different. You know, we're not little mice, and we're definitely human. So the body might be a little different in how they react to things. So the more therapeutic approaches we have, whether it's gene therapy or gene agnostic or gene editing or whatever have you, the idea is that whole idea of shots on goal really increase our chances. And what a great place it would be if you had multiple different therapies for the same indication, and then you can choose and pick for what you think is best for you at the stage of disease that you have, and what you want to do when it comes to delivery of that. So it really gives folks a lot more in terms of choices of what they can use. So obviously that would be a great place to be. Right now, there's really only one gene therapy for inherited retinal disease, and obviously that's like Luxturna, and obviously that's for a very small portion. But you know, those people that have that have, for the vast majority of them, have had positive benefit. So,
thanks for that.
Yeah,
perspective.
I might add too that, you know, as I mentioned, not all of us know what our causative mutation is, and so some of the approaches do rely on knowing that information, but there's also a lot of approaches that do not rely on that. So by having different ways of thinking about different types of therapeutics, some that are dependent upon knowing your gene, some that are not relying on that piece of information, allows us to think about treating the masses of those that have been diagnosed with an inherited retinal disease.
Right. Good points. We got a question about diet, and I think I would like to at least start answering that question about diet and retinal disease. I think it's an important question, and then let our panelists chime in if they'd like to. So what we generally say for people with retinal diseases, whether it's AMD or the inherited retinal diseases, what's good for your body, what's good for your heart is good for your retina. Lots of leafy greens and vegetables, colorful vegetables, fruits. While it hasn't been demonstrated definitively, the omega-3 fatty acids are thought to be good for your retina. Studies haven't shown dramatic results at this point, but it's a good idea to get your omega-3s from cold water fish, or you can supplement with them. If you're a vegetarian, you can get omega-3s from walnuts and some other sources, so that's pretty good. A researcher named Elliot Burson, those of you that have been with the foundation a long time are probably very familiar with Elliot. He passed away a few years ago, and he was actually our first researcher. We founded the Berman Gund Lab just after we were established as a foundation, and it was Elliot who led that lab. But anyway, Elliot was a big proponent of vitamin A palmitate for retinitis pigmentosa as a dietary supplement. And he also combined that with DHA as well as lutein. And he did clinical studies that we funded. Now, the results of Elliot's trials were not overwhelming. The improvements for patients was relatively modest. But he really proposed that therapy approach for many of his patients. I need to say there are many clinicians who don't believe in vitamin A therapy. And at the end of the day, it's really up to you as a patient and your doctor to understand and evaluate whether vitamin A therapy is the right step for you. Now, vitamin A therapy involves the use of vitamin A palmitate at a dose that's much higher than the normal vitamin A you might supplement with. And we actually have a guide on our website that can tell you more about considering whether vitamin A therapy is right for you. But it's something you really need to check out with your doctor. And if you go to our website and search on vitamin A, you can bring up an article about that and upload the document. And actually, as a follow-up, I'm going to make sure we disseminate that information when we disseminate the recording of this video. I think that would be a good idea. Now, one thing I want to emphasize for people with Stargardt disease or Best disease or other macular conditions is they should not even consider vitamin A therapy, because that could actually make things worse. So vitamin A therapy is for people with the outer retinal degenerations like RP and Usher syndrome. So I just wanted to emphasize that. Now, I want to get back to the questions about clinical trials. Those are always common questions. And one general question that we often get, and I think it's a good one, is how do I know what trial to enroll in? And obviously, if it's for a specific gene, you need to know what your mutated gene is so you can fit the trial criteria. But for trials that you do meet the basic criteria, how do you know if that trial is right for you? And I'll open that up to all the panelists if somebody wants to jump in and address that.
Hey, Ben, I think I'll take that first.
Okay.
So I think your first goal would be to have that discussion with your physician or with your eye care doctor. Having that discussion and that openness helps you better understand the condition that you have. And your doctor can also help you follow up on the kind of gene mutations you have. I know the FFB, or the Foundation Fighting Blindness, has the opportunity to allow people to check up on what gene mutations they have. And so if you know your gene mutation, or if you understand what your real condition is, then you can go to clinicaltrials.gov and check out on what trials are going on there. I think some of them will tell you what the inclusion criteria is and what the exclusion criteria is. Again, you would need to have that discussion with your doctor just to have some general sense of what the trial is about. And then it's okay to reach out to the trial administrators, and then usually they will guide you on what the next few steps are to help you participate in the trial.
Excellent answer.
Yeah, I definitely totally agree with what Bright just said. I did want to just mention one thing on clinicaltrials.gov, is that just because it is on clinicaltrials.gov doesn't mean it's necessarily been approved or vetted by any agency. So it's simply a way of posting that. So even when you go on clinicaltrials.gov, you do need to be a little careful as to make sure that you know who the sponsor is and things of that nature. So it's a good place to start your search and then ask questions after that with your healthcare provider and with your patient advocacy organizations, such as FFB. So, but it's a great resource to really understand what is out there. But once you find that, you definitely want to do a little more investigating as to, who's running this trial and things of that nature.
Right. Excellent point. And one thing I'd like to add is, if you feel like you look at a listing on clinicaltrials.gov and you think it might be a trial you're interested in, you reach out to the trial sponsor, the coordinator, and have a discussion with them about what the trial entails, how many visits you have to make, what the risks might be, anything else about the trial. So participating in a trial is a journey. You don't just suddenly sign on the dotted line and you're in. You really have to go through a process with the trial sponsor. And there's nothing wrong with just reaching out at first and asking some questions to get a sense of whether you want to move forward. So I think that's always a good thing to do. I know I get a lot of questions about clinical trials, about whether something is appropriate for a person. And often I just say, well, reach out to the sponsor and have a conversation and see how you feel about it. So,
and there's this process that's very important whenever you look into that, or when you're about to enter a trial, and that is the informed consent form process. And that process really should give you all the information of the trial itself. What are the potential benefits? What are the potential pitfalls and side effects and potential negative things that can happen? And that should be something that you and the principal investigator or his or her designee will really go through and take their time to do that. I know that at times we mentioned that the informed consent took longer than the actual surgery in our trials, because that is a time where you really get to ask those questions, and you really want to get a good idea of what you are now saying that you're going to consent to. And the reality is, you have that option as a person in a clinical trial to really withdraw at any point that you feel uncomfortable in moving forward. So that is a very important part of the clinical trial process, is that informed consent process.
good point. Good point.
Yeah, Ben, I also would like to add about reaching out. And reaching out to the sponsor is something we want the patients to be very comfortable with, and asking questions about what things they would need to know about the trial before they make a decision. And it's if my experience with AGTC, I would like to share my experience with AGTC, is the company or the trial sponsor, it's very focused on the journey of the patient as well. And so sometimes the sponsors would go every way they can or within their reach, just to make sure that any patient who is interested is properly assessed and so on, to be able to participate in the trial if they are eligible. So reaching out is really key. If you think you see a trial, you discuss it with your doctor, and you think in any way you are interested and might qualify, please reach out to the sponsor, and they'll be happy to guide you through the process.
Right. Excellent point. And I know that your team at AGTC, if people are interested in the Xlrp or achromatopsia trials, gene therapy trials, you guys are great in terms of helping people understand what the trial entails and what the journey is like. So thank you. We're getting close to winding things down. We have a few minutes left. And one topic that we have not really addressed at all, it's really worthy of a webinar unto itself, is genetic testing. Because genetic testing is really an important part for most people in their journey with an inherited retinal disease. And so I'm going to first point that question to our Chief Scientific Officer, Claire Gelfman. And I guess it's just a request for you to elaborate on it. It's not really a question, but tell us about genetic testing and why it's so important, Claire.
Yeah, no, thanks. So, I mean, obviously, when an individual goes to their retinal specialist and they walk out of that room with a diagnosis based on your clinical presentation, you know, one of the obvious questions is there a genetic basis for this information that I was just given? And the way to answer that question is to get genetic testing. And it's obviously important because once you know your specific mutation, then it opens up you to be a potential enrollee in a clinical trial where individuals with that specific mutation are being evaluated. Now, many individuals go through that process and come out of it not knowing. That doesn't mean testing was a failure here, because you still, so I should mention that through our registry program, when a physician, when your ophthalmologist gives you that diagnosis, they can put in a request, an order for genetic testing. And that genetic testing is actually free of charge through our open access program. And when those test results come in, you also have access free of charge to a genetic counselor to review the data with you, because it's a wealth of information. And then all of your information then gets deposited into our registry. And whether or not you know your specific mutation through your testing, by being now a member of this registry, what it puts you in a position is to get a call from companies through us for entry into clinical trials. So we work with a lot of different companies that use our registry to enroll individuals for their clinical trials. And whether, even if your mutation is not known, if you have a diagnosis of RP, for example, there are these mutation agnostic therapies that are looking for subjects for their trials based on that diagnosis. So again, whether or not your testing provides a definitive answer, you can still, by being a member of our registry, be in a great position to be called on for clinical trials. So the testing is very important. And I should also mention that it's really evolved through the years. You know, way back when, we didn't, you know, the early funding of FFB was about the identification of genes that, when mutated, caused inherited retinal disease. Well, now we've identified, you know, 300 or so of them. So now it's all about how can we use that information to come up with the best therapies? And that's a lot of what we've talked about today. So I really feel strongly that testing is important. And to get, if it's been, you know, many, many years since you've been tested, it's actually worth being retested because the methodologies are better, the number of genes we can test for is better, and having your information then in our registry puts you in a position for enrollment in a potential clinical trial. And I should also mention too that we and others use our registry for other things too. You know, it's very important that in defining the best endpoint to define success for a trial is based on the patient journey and how the mutation or how that diagnosis is actually implementing your day-to-day living. And we and others use our registry to survey individuals in order to learn about what that journey is like. And those endpoints that creep up, we are in a position to work with the FDA to hopefully have those as part of endpoints. I mean, that's a big process right there. But it's one thing to, you know, right now, vision lines on an eye chart, you know, are one way that is that's a sort of an approval endpoint. But we also want to learn more about the patient journey in order to work with the FDA to incorporate other endpoints. And a lot of information comes from individuals who had entered our registry, and they've gotten there through the genetic testing process.
Thanks, Claire. Excellent points. And I want to mention that the Foundation's no-cost genetic testing program is available to inherited retinal disease patients who have gotten a clinical diagnosis of an inherited retinal disease from their doctor, those doctors in the US. And it's the doctor that orders the test. A lot of people contact us wanting us to order the test. You have to ask your doctor to do it. And your doctor does it online. And you can learn a lot more about our no-cost genetic testing program by just going to our homepage, fightingblindness.org. And at the top in the center is a genetic testing tab. You click on that, and you can get a lot of great information about our genetic testing program. And again, it's available to any person in the US who's been clinically diagnosed with an inherited retinal disease. If you've gotten testing, received testing with a large panel over the last 4 years or sooner, you have to wait until that 4-year period has completed because you've obviously received testing relatively recently. But once that period has concluded, then you're eligible for our program. And thus far through that program, we've tested somewhere in the neighborhood, or I should say our partner Blueprint Genetics has tested somewhere in the neighborhood of 10,000 people. It's been a big investment for us, but a very worthwhile investment. And along with genetic testing in this program, you get no-cost genetic counseling from Informed DNA, or if your clinic has a genetic counselor, they can provide it as well. And genetic counseling is a really critical part of the process because the genetic counselor is going to help you understand what those results mean for you and your family, if there are other family members that are at risk. And if you didn't get a definitive result from genetic testing, and that happens in about 30 to 35% of cases, the genetic counselor is going to help you understand the path forward, what the next best steps are for you in your journey and pursuing additional testing potentially. So again, visit fightingblindness.org. Right smack dab at the top in the middle, you'll see the genetic testing tab, and you can learn more about the program. So we have just a few minutes left. I was wondering if any of our panelists had any additional comments to make before we officially conclude. First of all, I want to thank all of our panelists for their outstanding presentations. This was a great webinar. Dan, Bright, Claire, wonderful job. Dan Day, thanks for kicking things off. And if you guys have any final thoughts or comments, please share, and we'll go from there.
Just want to express
I just want to thank everybody.
Yep.
Go for it, Dan.
No, I just want to echo the same thing. It's been a pleasure, and obviously, if there are further questions or something, I'd be happy to answer those offline as well. So.
Okay.
Yeah, I really appreciate, the great attendance we have here today. We're here for you, want you all to know that. And again, feel free to reach out for any additional questions.
And I'd like to thank the audience very much, and to say that we are willing to be a part of their journey, and they should be open to reach out if anytime they think they can make it into any of our trials. Thank you.
Thank you. Thank you, panelists. And I want to thank the nearly 800 participants for the webinar through Zoom. I'm sure there were many more through Facebook because we showed this on Facebook Live. I didn't go to Facebook to see what the activity there was. but for everybody, we appreciate you taking time on a Saturday to learn about the research. And at the end of the day, it's the people who support our mission, many of you out there who volunteer and donate and do vision walks and work in your chapters, that makes all this research possible, or much of this research possible. So thank you. It's been a great 50 years, and we're doing our best to shut our doors down when we've eradicated all the retinal diseases, so we don't have to continue this challenging fight. So to everybody, again, thank you. Have a great rest of your Saturday, and we look forward to seeing you at the next event.
Thank you.
Thank you, Ben.
Thank you.